Hi, good afternoon. I hope everyone enjoyed lunch and the terrific posters that we had today. It's a tremendous honor to be introducing our featured speaker for the Body and Cancer session. Briefly, Dr. Gilles obtained his PhD from UC Davis. He went on to obtain a postdoc fellowship at Yale. He is the chair of the Department of Cancer Imaging and Metabolism. He's the director of the Center for Excellence in Cancer Complexity. He's the vice chair of research in the Department of Radiology, and he's the scientific director of the Small Animal Imaging Laboratory at the H. Lee Moffitt Cancer Center and Research Institute in Tampa, Florida. In addition to authoring over more than 240 publications, he's received numerous national and international awards for both his research and for his teaching. Dr. Gilles' vision for the Moffitt Imaging Initiative is the development of new applications to diagnose, predict, and monitor therapy response using non-invasive imaging, which is very near and dear to our hearts at the Translational and Molecular Imaging Institute. So his work spans both molecular and chemical, and from preclinical to clinical work, and he also leads a very robust training program as well. So it's a tremendous honor to be presenting him. He'll be speaking today on the MR imaging of tumor habitats.
Thank you very much. And first of all, I have to apologize not to be there. I just have one word to say, and that's Delta Airlines. So I was at the airport at 5:45 today, and they canceled this flight after being there 6 hours yesterday before they canceled the flight. So I have made an effort to get there, and I really apologize for not being with you. So I do, yes, I want to talk about tumor habitats, and hopefully by the end of this talk, you'll know what habitats are and why they might be of interest. First of all, I want to thank the group that's working on this. It's a very multidisciplinary group. This is the entire Physical Sciences Oncology Center at Moffitt that's made up of two departments, the Imaging and Metabolism departments and the Mathematical Oncology departments. I also want to just give a little advertisement for a meeting that we have that uses extracts quantitative features and converts images to structured data. It's called a process called Radiomics, and we meet every year at the last week of October in Clearwater Beach. And if you have an interest in joining us this year, just send me an email and I'll get you on the mailing list. So this shows the concept of where, well, it's not the concept, it's our roadmap of how we envision patients should be treated with cancers. And it shows down here in the right-hand corner where imaging of habitats and radiomic imaging fits within this whole paradigm. So we like to get from every patient as much data as we can. So it's not just simply, we don't do gene sequencing and pick a drug. We take the gene sequencing, we do pathomics on the histopathology, oftentimes with immunohistochemistry. We take a patient's history into account. We do liquid biopsies if we can get them. We have lab data, molecular imaging, and radiomic imaging, and we combine those using a mathematical model to predict where that patient is, where that tumor is, what it is, and where it's going. And these models are actually nonlinear models, much like predicting hurricane paths. The difference is in predicting a hurricane path is you're just a passive participant in this, but in cancer, we have the possibility of affecting the trajectory with therapy. And so we apply this therapy, and most importantly, we can collect these data here at the bottom longitudinally to update the model to tell us whether or not we made the right call. And this is a very, very important component. Notably, when we do pathology and genomics, that sort of sets the ball rolling, but it's only really with imaging and liquid biopsies combined that we can monitor the progress of the patient in response to therapy. Now, this looks like a nice picture, but we have concrete examples of how this is being put to practice in myeloma and in prostate cancer, and I'll talk about one of those. a little bit later at the end. So this is the motivation behind what we do. And the motivation is that, we spent a lot of effort in cancer research. We're developing targeted therapies, but they've been relatively disappointing in the clinic. Very few durable responses have been seen. Even when we have a great drug and a well-validated target, this is non-small cell lung cancer, breast cancer, melanoma. Doesn't matter what it is, when you start treating with these targeted therapies that interfere with signal transduction, we get a few months of benefit, and that's all. The problem is shown here, and this is an in vitro study where we took these EGFR mutant non-small cell lung cancer cell lines, here PC9, and we treat it with gefitinib, which is basically a sister to erlotinib, which cost $12 billion to get approved. And in culture, we see that this is a very powerful drug. It's got a very, very high avidity for its target. It's low nanomolar range. It can kill cells that have this mutation. But when you look at the clinically relevant dose of 1 micromolar, we're only killing about 92% of the cells. And so this is in a cell culture. The cell culture is supposed to be homogeneous, but even in the cell culture, we only kill 91% of the cells, and then we grow those cells out again and treat again, and we only kill about 30% of the cells, and so on and so on. So in just one drug holiday, we've selected for pre-existing cells that have a resistance mechanism against this drug. So the lack of a cure that we're seeing with targeted therapies is due to the emergence of these pre-existing resistant populations. Dobzhansky says many years ago, the father of modern genetics, molecular genetics, is that nothing in biology makes sense except in the light of evolution. And we have to agree with them. So the reason targeted therapies are failing is, one, because of evolution. And this, it's important to talk about the concept of evolution, or the mechanism of evolution, because it requires two components. You need selection and you need diversity. So in other words, you have to add a selection pressure onto a population that has diversity before you can hope to have any type of evolution taking place. And oftentimes that selection pressure is ignored in modern discussions of evolution in cancer, but it's absolutely critical. And selection is taking place either through habitats that pre-exist in cancers or through the therapy that we impose. And when we impose that on a diverse population, we're selecting for cells that have the phenotypes, not the genotypes, the phenotypes that will allow them to grow. The other reason why targeted therapies fail is ecology. Tumors have diverse habitats within them. And we can hope to see these and image these with multiparametric MRI. We also know that tumors are very heterogeneous genetically. So if we take biopsies from different regions of the tumors, what we'll find are different clades and clones of cells. And these are probably selected by living and existing in these different habitats. And this is something that we're trying to define. But if you look at the lower figure here, this is basically what a tumor looks like. It's mostly a desert. But you have some blood flow, you have some water flow, which does increase the density around the water, but generally speaking, it's pretty much of a desert. And so it's very easy to imagine that these different habitats are going to respond differently to different therapies. So tumors are not homogeneous, well-mixed systems. They're complete ecosystems comprised of habitats, and each with their own local selection pressure and phenotypes. So our first clue into this was a few years ago. One of my talented postdocs, Olia, we were doing radiomic feature extraction of non-small cell lung cancers. And what I mean by radiomics is that we take images and we extract from the regions of interest about 300, 400, 500 different features that describe that tumor, numerical features. And she created this feature called Entropy. And she said she noticed that in some cases, patients had the same entropy across the whole tumor, as shown here on the top. But in other cases, patients had very low entropy in the core of their tumors and high entropy at the rim. And so she decided to take that and distinguish or disambiguate the core and the rim as different regions of interest, and take the difference between the entropy in these two regions as a feature in itself. And you can see in the lower one, we've got a larger difference than we do at the top. And when she did that, she found that those that had the largest difference had the worst outcome. And even this one at the bottom, if you look at these two, a radiologist will look at these, and because this is attached to a pleural wall, you would expect it to have a better outcome, but not if it's got a very flat entropy across it, the whole tumor. So this is our first clue that something is different about the edge. And we followed this up, the tumor core and the tumor edge being different habitats. We followed this up microscopically with a very talented scientist, Mark Lloyd. And here you can see an invading ductal carcinoma, invasive ductal carcinoma of the breast. And you see, I think you can see pretty clearly that up here in the upper right, we've got a high infiltration of tumor cells that look fairly homogeneous here. We've got quite a few lymphocytes, but then as you get closer to the edge, this lymphocytic infiltration has really increased quite a bit, and especially around the edge of the tumor. And then you have the stromal fibroblasts out here. So the edge here is quite different than the core. And so we wanted to look at this and determine what the phenotype was of these different cell types. And so we took regions from whole mount biopsies from the edge and from the core, and then we stained them with immunohistochemistry. And what we see here is a compilation of a lot of work, and basically the same pattern that we saw over and over and over is that in the core, the cells are denser, fewer lymphocytes than at the edge. The edge has a high expression of carbonic anhydrase 9, which is supposed to be a hypoxia stimulated marker, and I'll tell you about that in just a minute. The edge has lower expression of carbonic anhydrase 12, which is not hypoxia upregulated. More proliferation at the edge than in the core. More apoptosis, cleave caspase 3 in the core than the edge. GLUT1 is also a canonical hypoxia upregulated target. It's associated with a high amount of glucose metabolism and fermentation. That's at the edge, not at the core. HIF1 alpha, the hypoxia-inducible factor, higher at the edge, and it's more vascularized at the edge, indicating that it's not hypoxic. It's well vascularized. So the stroma into which the tumors are invading is well vascularized. The stroma is not hypoxic, yet we're upregulating all these hypoxic markers in a condition that we think of what we're calling pseudo-hypoxia, because the cells are secreting or manufacturing hypoxia-related proteins, but they're not hypoxic. Carbonic anhydrase 9 is of special interest to us going forward. We want to look at it therapeutically because it does, one of the things that it does, it acidifies the microenvironment. It has a very low pK, and when it's presented with CO2, it converts it to an acid. And then therefore acidifies the extracellular microenvironment. This could be an entirely different talk that I could give, but this is something that we're really quite interested in. So the rationale, what I've sort of established so far is that evolution requires this microenvironmental selection of populations that have phenotypic diversity. This phenotypic diversity is apparent at the microscopic level. These microenvironments can be imaged non-invasively as habitats, and that's what I'll be talking about for the rest of this talk. So we hypothesize that these habitats should be spatially related to the cells and tissue phenotypes that they contain. And what that means is that there's a relationship between the genomics and the habitats, and that these habitats can be used to monitor and guide therapy while it's ongoing. And this is all work in progress. So here's a shot, just a screenshot of what we do with radiomics. We get the patient images. We can either take the whole region of interest, or we can define subregions that are called habitats. And then from these, we extract the structured data and combine that with clinical and genomic data to build predictive models. With MR, we have the choice. We have an abundance, a wealth of information that are gathered from MR images, typically as standard of care. Two of the most common ones are diffusion MRI and dynamic contrast enhanced MRI, and I'll talk about those in just a second. I know I don't have to introduce it to this crowd, but there might be some folks in the audience that don't use these. And then we have a number of other techniques that we can apply. The process that we go through in habitat imaging is to acquire these multiple parameters in a single exam and then combine them in strange and wonderful ways to cluster them into habitats. So diffusion, just to remind you, if you don't use this, we impose two orthogonal diffusion gradients separated by in time, and during this time, water molecules have a chance to move, and if they do move, we lose signal. However, in true diffusion, there's no barriers. However, in tissue, there are barriers to this motion, specifically cell membranes, and so intracellular water is not as mobile as water that might be freely diffusing. So what we get when we measure the diffusion images, when we measure the diffusion coefficient of tissue, is an apparent diffusion coefficient that is related to the cell density. Here's an example of just an axial image of a patient here, and you can quantify these numbers, these diffusion numbers, in millimeters squared per second. And you can see here that the metastases, or the tumors, generally have a much lower diffusion coefficient than other tissues because the cells are so dense within tumors. Dynamic contrast enhanced is basically a T1 image, a T1 method, where you acquire time-dependent change in signal intensity following injection of a contrast agent. And I think what's important to note in tumors is the heterogeneity of how this enhancement occurs across the tumor. So you can't basically draw an entire region of interest across a tumor and gather any meaningful information. You have to be able to process these data spatially located. So here's just some small ROIs, but right now, typically we do these diffusion measurements on a pixel by pixel basis, and you see, 4 completely different types of enhancement patterns. And we know that this enhancement pattern here is probably the worst prognostically, whereas these other 3 are much more benign. So here's an example, just taking standard of care image data that we can pull off of our medical records of sarcomas. And we take, in this case, we took just a post-contrast T1. Really, we're taking the post minus pre. And if it's highly enhancing, we put it over here. If it's lowly enhancing, we put it over here. And then we get a T2 measurement, quantitative T2, separate those into low and high. And then we color code these 4 habitats accordingly. Now, this is just a mathematical operation. And there's no reason to expect that these things would cluster spatially together, because we're not using any spatial information in this model at all. But I think you can see on the upper right that, you know, these habitats do, these do cluster spatially together. And then this is why we're prompted to call them habitats, because they're reflecting spatially distinct, physiologically distinct portions of the tumor with different selection pressures. And importantly, in sarcoma, you know, with over 25 different histological subtypes, there's a lot of heterogeneity in this disease, but across all the sarcomas that we've looked at, they have these habitats. And interestingly, these habitats are prognostic. So we can use the pre-therapy, pre-neoadjuvant therapy to identify patients that have pre-existing metastases. We can use them to predict overall or progression-free survival. So of all the covariates in this study that we did, which is published in the previous slide there, you can see in a medical imaging journal, the habitat, this yellow habitat, if it exists in more than 18% of the volume, had a very, very poor prognosis. And this is T1 high, T2 low. So T1 high means probably either pretty well perfused or hypoxic, but T2 low means it's probably very high cell density, because T2 is also related to cell density. We're just guessing, but we know that this combination here is a very strong prognostic indicator. So we've been taking this a little bit further. Dr. Raghu has been looking at this and has been able to come up with a new schema to define more habitats based on T1 differences and flare and T2 and diffusion. And results of that are shown here. So here we're basically taking, you know, 5 different pieces of information here, combining them, in this case, using Otsu thresholding, and are able to generate this map here of a glioblastoma. Notice that we have, on the previous slide, we have 12 habitats to choose from, but in reality, we only see about 6. And some of these are fairly easy to define: tumor enhancing, tumor non-enhancing, edematous regions, CSF, that's pretty clear, white matter, that's pretty clear, and this thing that we call immune infiltration here. Most importantly, I think, for this concept of habitat imaging is that we can then use these habitats to monitor patients' progress as they get treated with therapy. So this is this same patient, she was treated with nivolumab, a checkpoint blockade, and here, and you can see the evolution of this immune infiltrate. Hold on. Okay, this immune infiltrate, and then it resolves while she's still on therapy, and of course she progressed at this point. Even before therapy, though, these are TCGA data. Even before therapy or surgery in these patients, we can predict who's going to progress very rapidly and who's going to be less rapid in their progression using a 400-day cutoff. And you can see this is the fast group here, which has a low T1 and a high flare, whereas the slower group had a higher T1, post-contrast T1, and a more moderate flare values. And these are pixel by pixel. So to process these data, we have to do a lot of interpolation to make sure they're on the same grid. So I said earlier that we know that cancers are genomically heterogeneous. This is true for GBM as well. There's a paper where I wish they would have actually, that this image here actually bore some relationship to reality, but it doesn't. So they have no idea where these are coming from. There's a couple of things that are important about this slide. You know, one is that they build this, they build an evolutionary tree, but there is no evolution here. This is just a bunch of genes. You can follow their, their timeline. You can follow their, which one came first, but you can't infer any evolution at all. The other thing I think that's important here is that of all these biopsies, almost all of them were proneural, which is a relatively slow growing and or less aggressive form of GBM, but one of them was from the mesenchymal subtype, which would be the biopsy that would guide therapy. So basically, out of all these biopsies, they had 7 chances out of 8 of getting it wrong. So if these areas, if these genomic landscapes exist in specific habitats, then we can use the habitats to identify areas that should be biopsied, because that will dictate the choice of therapy. So that's a big question for us right now in GBM, is how these habitats that develop as the cancer grows, how they relate to these genotypes. So the rest of the talk, I'm going to talk about how we're going to try to unravel this. One, we're doing it preclinically, and we're using breast cancer models. This is Bruna Jardim. The one reason for doing it preclinically, although they don't have time to evolve all these habitats, is we can do just a tremendous amount of imaging on any one mouse. We can get really dozens of images that we can combine. So and a lot of dynamic and quantitative data as well. So we've done this in a number of mice, and importantly also about the mice is we can then use the T2 map to create an isosurface, and that isosurface is used to plan the printing of a 3D mold. And you can see the mold right here. And then we can isolate the tumor from the mouse. We paint it just to get oriented, put it in this holder where we do a CT scan to make sure that it's fitting correctly. And then we slice it, put it in a cassette, and stain it. And this is fixed, by the way. This is fixed by this point here. So, and we've been able to generate or use some fixatives that don't result in a lot of shrinkage. So our registration is pretty good. Here's how we do the registration. So you can see the different slices of a mouse on the left there, and 15 different slices. And in the middle here, I show the tumor itself, the isosurface of the tumor. And these planes, these colored planes, show you the location of those cut planes, those 2 millimeter slices. And then on the right here, you can see that's color coded as well. What we've been able to do is take that plane, identify what the cut surface was, and from the MR image, and then project that back onto the H&E section. And I think you can see across all the slides here that the registration is pretty good. So we're actually coming up with numbers that are leading us to believe that our root mean squared error is about 3/4 of a millimeter. So I think we're pretty good in terms of doing this, of registering. And then the idea then is to take the MR images and project them back onto this histology to train this, use the histology to train the MR images to identify specific habitats within this domain. And we're getting very close, at least in the training set. We haven't done the test set. The test set, of course, is to take the MR images and predict what we're seeing in the histology, basically to do histology by MRI. Some of the things we're interested in, like I talked about before, is carbonic anhydrase 9, pimenidazole, which is hypoxic, a hypoxic marker. And I think you can see over here on the right, some justification for my claim that the carbonic anhydrase 9 is not just in hypoxic areas, but it's also being constitutively expressed elsewhere. Right? And that the table at the bottom shows that. And this is just an example of the histology, the histological habitats that we can see, and the MR visible habitats that we're trying to relate to these. So that's a preclinical approach. We're also doing a clinical study with Radka Stoyanova at University of Miami of men with prostate cancer. And this is a cohort of men who are under active surveillance where they get transrectal biopsies, core biopsies annually. And importantly, we have MR guided ultrasound system. So MR ultrasound fusion, the biopsies are ultrasound guided, but we can fuse the MR images with the ultrasound to know where the biopsies are coming from. And we get 14 biopsies per patient per session. And these have multiple H&E cores within them. So the idea is to do the genomics, do the pathomics, register them back onto MR and integrate. And it's coming along quite well. So we've accrued about 30 patients on this study. The important thing is this is a commercially available system. It's not perfect, but it's right now good enough for the use that we want. We have this URANAV system, which fuses the MR with the ultrasound, and then we have the ultrasound guided biopsy. An example of that is shown on the next slide here. Here's a man, two different slices, 3 different imaging routines, a T2, ADC, and DCE map. And you can see this red area here is a hypointense on the T2, hypointense on the ADC, hyperintense on the DCE. These other 2 down here at a different plane, you can see somewhat hypodense in the green area, not so much on the ADC, some hyperintensity on the DCE. In the blue area, hyperintensity, but not so much. It's in the central area of the gland. So we mark these as regions of interest that should be biopsied. And over on the right, you can see that this, well, in section B, we have the transrectal ultrasound, where we did the ultrasound guided biopsies, and C shows the biopsy tracks and the histology that came out of these 4 areas, the 3 areas, the green, the purple, and the red. And the red turned out to be Gleason 7. The other 2 were Gleason 6. If a man is on active surveillance and shows a Gleason 7, then he'll progress onto therapy. So that's identifying the area that we want to biopsy. So we get the biopsy that's going to dictate treatment. And, you know, like I said, we're doing genetics, but I'm not interested in genetics. I'm not going to talk about that. I'm saying that somewhat tongue in cheek, but if I was there, we could talk about it some more. So once again, getting back to this paradigm, this is something that we're trying to employ. And I'll give you an example of just one thing that we're doing along these lines in the last few minutes, and that's in prostate cancer. Once men have progressed onto treatment, they're treated with Lupron for chemical castration. And when they progress off of that, one of the reasons they progress is Lupron inhibits in the LHRH pathway, inhibits the production of testicular testosterone. One of the reasons they progress on Lupron is the tumors themselves start to make testosterone. And this can be inhibited by a new drug called abiraterone. So men, you know, go Lupron, go abiraterone, and the concept that we've talked about for many years, so you can see been doing this since 2009, is that tumors have different cellular phenotypes. And in prostate cancers, castrate-resistant prostate cancers, there are 3 general types of cells. You have androgen-dependent cells that don't make testosterone. Those are cheaters. You have androgen-dependent and androgen-producing cells. And then you have androgen-independent cells that might have an androgen receptor variant. that doesn't need androgen in order to grow. So our idea, and this is really the idea of Bob Gatenby, my colleague, I'm stealing his thunder here, but only because it's so interesting, is that when these tumors present, the androgen independent cells are less fit. There's a cost to this, to androgen independence. If in the presence of androgen, an androgen dependent cell is much more fit than a androgen independent cell. So it's a minor population. And we want to keep it a minor population by maintaining the other two populations at about their present level, at a subclinical level. So the idea is to treat periodically with abiraterone. And so here you see these top two are simulations where you treat and then you let the tumor grow back, you treat, let the tumor grow back, et cetera, et cetera. Or if you have a very fast growing tumor, you have to treat much more quickly. And what we've seen with men on this trial is that each man has his own periodicity. So here's one patient, and he was treated, and his PSA just declined. And then it took almost a year or over a year for it to come back up again to get retreated. Whereas we've got this other guy who's just every few months, his PSA is shooting back up again. So we're adapting the therapy based on the patient's response. If they, we don't treat them if they don't require it. If their tumor's responding, we take away treatment. This is the cohort so far, 12 men, and all of them have, none have progressed, and all have gone well beyond the control time to progression of 9.3 months. So these guys progressed on imaging, but they haven't had a biochemical recurrence yet. And these men have voluntarily on their own discontinued Lupron, which they hate. So I think this is a very enticing justification for this model of how we want to conduct trials going forward. So my summary of my whole talk is that tumors are not homogeneous, well-mixed systems. They're complex ecosystems and must be characterized as such. And Albert Einstein once said, you should make things simple as possible, but not simpler. And I think that's exactly what's going on in cancer research today. Thank you.
Thank you very much for that very interesting talk. Would like to open it up to any questions. If anyone has any questions, please come up to the microphone so Dr. Gilles can hear you.
Oh, okay. That was a very interesting talk. Thank you very much. I have a question about the dynamic contrast enhanced T1 weighted imaging used to parse the tumors into 4 different categories of contrast enhancement. So my question is, to what extent is this information useful for understanding or motivating further research to understand the pathogenesis of these tumors? And to what extent is this categorization sort of a clinically useful black box that doesn't really help us understand the pathophysiology of the disease?
Yeah, that's a very good question. I think it's a mixture of both. I mean, I'm not afraid of black boxes, but I would like to know what's going on. Obviously, angiogenesis and perfusion has been very well studied in tumors for years. We've written extensively on this, not so much about angiogenesis and blood supply, but more on perfusion and the state of tissue that is subjected to intermittent perfusion. So we do quite a bit of in vitro work recapitulating those conditions to investigate what impact this has on the selected cell's physiology. And specifically, we're interested in the Warburg effect and glucose fermentation. But besides that, without getting into the cancer biology mechanism of this, it's got to work. You know, even if it's a little black boxy, we have to massage the image data to the point where it does work, where it gives the best predictive and prognostic features, and then we're going to let that tell us what's important and what isn't within these data that we're gathering. Does that make sense?
Oh, yeah, yes, thank you.
Yes, thank
Okay.
you. And I had one quick question for you, too. I was very interested to hear how this really integrated multi-parametric approach can really provide such robust information about different tumor habitats. And with respect to prostate cancer, because we see a large volume of patients here, has there been any interest in using this kind of integrated approach to try to detect or improve cancer detection rates? Especially for low-grade tumors like 3+3, just qualitatively, we're okay at finding these, but that's certainly one area for improvement in the future.
Well, I mean, I think our vision for, and one of the reasons we're really enthusiastic about the active surveillance cohort is that we would like to get to a state one day where the images, quantitative images, can identify when a man should and should not get a biopsy, and when he gets a biopsy, where it should go, you know, from what region it should be taken from. And, you know, I think that's a goal for a lot of people, but we're sort of obsessed on this application. So yeah, I think in terms of monitoring early stage disease, 3+3 especially, I think, you know, if you can show men that they don't have to get an annual transrectal biopsy, but they do have to get imaged every 6 months or every year, then you know, I think the uptake of active surveillance will be greater, and you'll see a lot more men with 3+3 in your clinic. Yeah. In terms of early detection, you know, in lung CT, low dose CT scanning and mammography, it makes sense to have a screening imaging test. I'm not sure if MR is ever going to be useful as a screening test, but you know, I'd love to be proved wrong. What I think is I think the circulating and tumor-derived DNA biopsy world, I think, is maturing very, very rapidly. And at least I know in pancreatic cancer, cervical cancer, ovarian cancer, I saw some data at the AACR indicates that, you know, that these are pretty much well on their way. If you have a positive test there, then a secondary screen would be imaging. I think, you know, the whole idea about screening is we need multiple filters to identify people with cancer. There's not one test that's going to do it.
Thank you very much, and thank you for the wonderful talk. We do have a small token of appreciation for you. Unfortunately, I know you can't see it, so I'll describe it for you. It's tear-shaped, it's beautiful, and it has your name on it, and we'll be happy to send it to you, and we thank you very much.
Thank you. Thank you.